This is chapter 15. It's dealing with the innate immune. The body has two different types of defense systems, or two subcomponents of the immune system. The innate is the first one of this. It's a way the body has of recognizing foreign cells, and then it will attack those cells. It can detect what's not part of you. The innate response is going to be the same whether or not, say it's tree pollen, whether E. coli, it doesn't matter. It is non-specific, and so it will attack it. The same methods, using the same type of things to attack those foreign substances. The innate is sometimes further divided in what we call the first line of defense and the second line of defense. This first line of defense, it can be a chemical barrier, it can be a physical barrier. It is a means of trying to prevent the foreign substance from ever entering your body in the first place. Can't get in, you don't have to deal with it. One of the major things will be your skin. Your skin acts as a protective physical barrier. You also have things like your mucous membranes as a role as a physical barrier in preventing anything from being able to penetrate. If we look at the skin, some of this may be a review if you've already had A&P1, but just as a reminder, the skin is composed of two major layers. You've got the epidermis, which is on the surface. It is multi-layered cells. The outer layers that you actually see are actually dead cells. What you're seeing is dead. And those cells are constantly being shed, removed, because the living cells are beneath them. They get pushed up layer by layer. So what you see on the surface is actually dead. So number one, a lot of pathogens cannot penetrate through these multi-layers. And even if they are on the surface of your skin, because those cells are constantly being shed or sloughed off, usually the pathogen is going to be sloughed off as well. There are cells that are known as dendritic cells that are in the epidermis layer that are their whole purpose is to carry out phagocytosis, meaning they will engulf, they'll eat up, chomp up, chew up any foreign substance. The dermis layer is beneath the epidermis. Between the two layers, there is a layer of collagen fibers that really help to, number one, protect the skin from abrasions, cuts, and everything, because anytime you break this barrier by a puncture wound, by a cut, that's an opening for, or a means or a way for the pathogen to be able to penetrate deeper into the body. So this just shows a scanning electron micrograph of what the surface of human skin looks like with those flattened, what we call squamous cells in there. In addition to being a physical barrier, the skin also contains chemicals that will work as a chemical barrier. You're producing sweat, and that sweat does contain some salt. Salt usually will inhibit the growth of microorganisms. There are other proteins that are present in your sweat that also act as bactericidal components. In other words, they will kill bacteria. And then you have lysozyme, which will also break down the cell wall. So all of those will work, are components in the sweat, and will work from a chemical means, preventing whatever the pathogen is from being able to penetrate into the body. Your oil glands, although they're helping to keep, hopefully, your skin nice and soft, that also helps prevent it from tearing, from drying out, and it will lower the pH of the skin a little bit. Most bacteria prefer a pH of about 7.5 to 8. So that's slightly alkaline. So if you lower the pH, as we've talked about in lab, that actually will help inhibit or prevent from growing. Mucous membranes, those will line all body cavities that have an opening to the environment, like your mouth, your nasal cavity, etc. They're also composed of different layers. The cells, once again here, tend to be tightly packed. They're continually shedding of cells. all work once again, like on the skin, to prevent pathogens from being able to enter in. And once again, produce various chemicals that will inhibit the growth of microorganisms. So this is just showing the respiratory system is lined with mucous membrane. So this is your way of protecting yourself. Uh, like I said, this is the first line of defense. There's a comparison between the skin and the mucus. Uh, it doesn't matter what the foreign substance is. Once again, it doesn't matter what type of, say, species of bacteria it is. It doesn't matter what kind of pollen it is. This is where the body's just recognizing self, you, versus non-self, which is not you. The lacrimal apparatus, a lot of people don't think about this, but it does play a huge role in the innate immunity. The lacrimal apparatus is what produces your tears and then drains the tears. Every time you blink your eye, your tears are flushing and washing over the surface of the eye. In your tears, there is this chemical, lysozyme, that will kill bacteria. The eyes are a potential entry point into the body. And so by having the lysozyme in tears and every your eye, then it will help to protect. So the lacrimal apparatus, actually your tears are produced up here in this close-up. You can see right here. You would blink your eye, those tears flush over the eye down here into the lacrimal duct. So that's helping to protect the eye. You have your normal microbiota. We have talked about this in the past, the normal microorganisms that are living on the surface and in you. That some of these, remember, are resident ones. They've been there since shortly after birth. Some of them are transient. They kind of come and go over the course of your lifespan. Now, these are going to be competing with any potential pathogens. Any microorganism has to have certain resources. They have to have nutrients, they have to have certain gases, they have to have water, excuse me, for survival. And so everyone's competing for the same resources. If your normal microbiota are there, those normal microorganisms that are there all the time, it's kind of like equating it to a very busy parking lot when we have face-to-face classes, in that there's only so much space. Well, there's only so many parking spots in the parking lot. If all the parking spots are filled, there's not room for anyone else to come in and park. It's just a space thing. There's no room. So if you have all of your normal microorganisms that are on the surface of your body, there's no place for the pathogen to set up shop, if you will. They would have to compete against the normal ones for those nutrients, etc. And so this is one of the reasons why I know in the past ones are asked, do you want to actually sterilize your hands? No, you don't. Are you trying, when you wash your hands, to kill everything off of the surface of your hands? No, you're not. You are just trying to wash off, essentially, think of it as the newcomers. Keep the regular guys there, but just wash off the newcomers. If you remove microorganisms that are naturally in there, they have a niche, they have their place there, you remove those, then that will provide an opportunity for the pathogens to set up. In the skin, the mucous membranes, etc., there are often antimicrobial proteins that there's a variety of these that will basically work against the pathogens as a means of protecting you. This is a table that is showing some of the different, what would be considered first lines of defense, different components of it, different things that may, in their secretions, have an effect on helping to protect you on this first line of defense. And it's set up by system. So such as in the digestive system, you've got the saliva that's naturally going to be kind of rinsing your teeth and your gums, your tongue, the palate, It does contain lysozyme in it, trying to kind of keep those surfaces clean from bacteria. And it goes through, there's the digestive system, as you can see, the reproductive and cardiovascular system. The second line of defense, this is when I've talked a little bit in the past about how you have this back and forth going on. It's like us against the microorganisms. So how are you going to protect yourself? Well, from our standpoint, we've got our first line of defense. We've got these physical and chemical barriers that hopefully will prevent the microorganism from ever even penetrating and entering into our bodies. It's like hot. You can't get past this. Well, sometimes the pathogens are capable of maybe producing certain, say, enzymes like collagenase that will break down the collagen fibers in the dermis layer that allows them to penetrate deeper into the body. So if the pathogen is able to pass that first line of defense, they're like, ha, we got past your first line of defense, and then we come back with, oh ho, we have a second line of defense now. So the second line of defense is still non-specific, so it doesn't matter what the pathogen is. It doesn't matter, once again, if it's a bacteria, if it's pollen. There's another way of dealing. You got past our outer perimeter, fine. We have another means of trying to combat this. This can be involving components of the blood, cells that are in throughout the tissues, etc. Some of the defense components of the blood, number one, you've got the plasma. Plasma is basically the liquid portion of the blood. It has electrolytes, it has gases, nutrients, proteins in it. It does contain some iron-binding compounds. Iron is definitely needed for metabolism. It's also needed by bacteria for metabolism. And then there are some antibodies that are found in the plasma, and a specific type of protein called complement protein. You also have the blood cells. Leukocytes, those are simply white blood cells. When you look at blood, you've got the plasma, and then you have what we call the formed elements. There's three types of formed elements. It would be your erythrocytes, which are your red blood cells. Their purpose is to carry the oxygen, carbon dioxide, CO2. You also have the platelets. They are involved with clotting of the blood. And then you have the leukocytes, or the white blood cells. So the purpose of the white blood cells is defense. They are defending against these foreign invaders. And you know how we like to divide things. So the leukocytes are further subdivided into two categories, either granulocytes or acytes. For the granulocytes, there are three different types. In agranulocytes, there are two different types. This picture here is just showing all the components of or types of the formed elements. So over here is the red blood cell or erythrocyte. Here are the platelets. As I said, those are going to be involved for blood clotting. And then these five are your five different types of white blood cells or leukocytes. These three right here, the basophil, neutrophil, and eosinophil, are your granulocytes. The monocyte and the lymphocyte are the agranulocytes. The agranulocytes, the three types are the basophils, eosinophils, and neutrophils. We can distinguish them when we do a blood stain in a dye as to, number one, you're going to look at not at the nucleus initially, look at the cytoplasm and see what color are the grains. And see also, number one, are there grains or not? If there are grains where it looks like someone's dumped sand in there, then that's going to tell you it's a granulocyte. The basophils, these are going to stain blue. Eosinophils are going to stain red or kind of an orange. Neutrophils are more of a purplish type of color to the granules. Neutrophils and the eosinophils, these both are involved with carrying out phagocytosis. The agranulocytes, it does not look like someone dumped sand in it, so you don't see those granules. And there's two main types, the lymphocytes and monocytes. The lymphocytes are going to be involved in the adaptive immunity, and that's what is going to be discussed in chapter 16. It also can, there are cells that will develop into what are known as natural killer cells, and those will be discussed a little bit later as well. The monocytes, when they're in the blood, they're considered initially monocytes as they leave the blood and can travel throughout the interstitial area that's between the tissues, the space between tissues throughout the body. They will mature and become what we call the macrophages. And they certainly are very good at carrying out phagocytosis. So it's kind of like they're on search for any of these pathogens or foreign substances that somehow were able to get past that first line of defense. The idea is that the macrophage will be playing a huge role in being able to detect them and identify them and then devour them. So over here are our granulocytes. If you notice, the coloration of the grains is different between them. And then you also look at the morphology or the shape of the nucleus. Does it have two lobes? Does it have multi-lobes to it? So you would be able to distinguish these by looking at a blood smear. Just to point out these little smaller, darker purplish blue color substances right here. These are your red blood cells. These are platelets. Over here, the agranulocytes, you can certainly see the difference in the cytoplasm, that it doesn't have all those grains in them. And the lymphocyte, the way you can distinguish these is look at the nucleus. The lymphocyte, the nucleus is going to take up most of the space of the cell. The monocyte, it has this irregular shaped nucleus. So you can do what was known as a differential white blood cell count. And that is when you are looking not only at the total number of white blood cells to red blood cells and looking at that ratio, but you are distinguishing now. Like I say, you look at it. How many lymphocytes? What's the percent? And you're looking at the ratio. What's the percentage of eosinophils? What's the percentage of lymphocytes? So as just examples that show you, if you have increased number of eosinophils, that those numbers are higher than what the normal ratio would be, that could be indicative of an allergy. So maybe right now a lot of you might have increased or elevated numbers of eosinophils with all this pollen. It could be indicative of a parasitic worm infection. Hopefully that's not the reason. If you have an increase in overall in the leukocytes and specifically the neutrophils, that's often indicative of a bacterial infection. If you have an increase in the number of lymphocytes, that's often indicating, okay, the patient has a viral infection. So the differential white blood count is not always ordered, but it can give you an idea of what is going on. Phagocytosis, this is what several cells are capable of carrying out. They divide it into different stages, as you can see. Basically, it is where it is able to recognize something as foreign and it. So here is your phagocyte. It could be a macrophage, it could be an eosinophil. Whatever type of cell it is, it could be a dendritic cell that I had mentioned, is that typically what happens up here, you have, in this case, your microorganism, whatever it may be. They often secrete certain chemicals that will attract the macrophages to that area. And oftentimes it may be you that's secreting the chemical that's attracting you to an area. When you have, say, a cut, part of the process, as soon as that tissue where the cut is has been damaged, it releases certain chemicals. Those chemicals will attract certainly like macrophages to the area, which are capable of carrying out phagocytosis. It's kind of like the body's way of saying, oh, we have a breach. We have a breach on this first line of defense. The skin has been damaged. We potentially have microorganisms, which are foreign, that may penetrate into us. So we need backup here. We need the cells that can carry out phagocytosis here to help engulf and get rid of these foreign invaders that have just penetrated our first line of defense. Chemotaxis is an attraction. Well, there's two different types of chemotaxis. There's positive chemotaxis and negative chemotaxis. Positive chemotaxis is when a cell is attracted to a chemical. Negative chemotaxis is where it's repelled, it's pushed away from it. In this case, your phagocytic type of cells are attracted to these chemicals, so that's going to be the first thing. Oftentimes now there are going to be receptor sites on the surface of the phagocyte where the microorganisms basically adhere or get stuck to the surface. Then it's going to ingest it. It's going to kind of wrap around it and engulf it. Now, once it's inside, it's kind of like, okay, I got you inside now. If you remember when we way back in the beginning talked about cells, you often have these little vesicles called that contain lysozyme. The lysozyme is a very highly digestive compound, like we just said. It will break down bacteria. This will be attracted to this vesicle that has the bacteria in it. So it's going to fuse to it, dumping out all that lysozyme in it, which is going to start to just dissolve and break down whatever was inside here, in this case a bacteria. The cell can reuse things, so if there are amino acids there, it can reuse it. Any remaining waste, it will eliminate from the cell by exocytosis. There's another way that eosinophils can attack cells. Oftentimes what happens is eosinophils, in terms of, remember I said they can also have elevated numbers if you have a parasitic worm infection. Helminths are worms, and so they're usually bigger than the eosinophils, so how is it going to engulf it? So what happens is the eosinophils, they will, now it's sort of a reverse thing, they will attach to the surface of the worm. And not just one, but several, so it's kind of like it coats it. And then it will secrete toxins. Those toxins will first weaken the helminth or the worm and eventually kill it. We also have these NK cells, or natural killer cells. They tend to secrete toxins also onto the surface of cells that have been infected with viruses. They can also recognize tumor cells. They are able to distinguish between normal body cells and abnormal body cells. Typically, when you have a cell that is cancerous, developing into a tumor, or you have a cell that has been infected by a virus, there are certain proteins that tend to be expressed on the surface that are not usually there. It's kind of like a little flag is put on the outer surface of the cell. And so the natural killer cells will say, oh, well, a healthy cell does not have this, so this is not normal, and so I'm going to attach to secrete the toxins. So that is its job, is to kill any abnormal cells. Neutrophils can also produce chemicals that are going to be killing abnormal around it. There are some other chemical defenses, and it involves what we call toll-like receptors, or TLRs. Once again, this has to do with different proteins, etc., that are on the surface of the membrane. Just be aware that they are there. We're not going to spend time on this. There are several different types of these TLRs that do play a role in recognizing your body recognizing foreign substances as being foreign. So there's several different ways of recognizing these interferons. These are proteins that are released by your host cell when it has become infected by a virus. And the purpose of the interferon is to help prevent or help stop the spread of viral infections within your body. That actually can cause some of the symptoms that are associated with a viral infection. But the idea is to help decrease the spread of the viral infection within your body. There's two main types of interferons, type 1 and type 2. Just so you know, this is another one of those lovely words that depending on where you work in the country, there may be different pronunciations. Some areas of the country will refer to these as interferons, which comes the first time that you hear it. When you have a cell that has become infected by a virus, such as up here, we have a virus that has infected one of your cells. One of the things that will happen is that viral infection will trigger activation of certain genes, which will release the interferons. At a later point in time, you're going to see what happens. The virus starts to go through its replication process. Meanwhile, the cell is releasing these interferons. These can be released and then go to an adjacent cell that has not been affected by the virus yet, and it's going to bind to certain receptors. And what will happen eventually is that when that virus tries to infect it, it's recognized as number one, this cell has a virus, so other viral particles from the virus standpoint will not infect it. However, the cell's going to eventually die, and ideally it will die before it can release new viruses. Now this is showing how an infected cell, without the release of the interferons, it goes through the viral replication route, produces all of these new viral particles. But the adjacent cell that had the interferon from this originally came over here, and over time, when this virus tries to infect here, it will not go through viral replication like what happened up here. Instead, what's going to happen is there are certain proteins that are called antiviral proteins, and they're going to stop and prevent the virus from replicating. So these cells will never produce new viral particles like what happened over here. So it's kind of a multi-step thing. Can the interferon protect the initial cell that was initially infected by the virus? No, this one is still going to produce new viruses. However, release the interferon and all the adjacent cells now can be protected. As I said, there's two different types of interferons, 1 and 2, and this gives some of the different comparisons between. All of this, remember, still is with your innate second line of defense. Complement. This is a whole series of proteins, and they numbered these. And just so you know, if you look at the numbers, you're like, this does not make sense. Somebody did not know how to count. They are numbered in the order that they were discovered. So it's not necessarily in the order that they are activated. The numbers have to do with when they were discovered. We're going to come back to complement a lot when we talk about the immune system. In a nutshell, what you need to know is when you see complement, this whole series of proteins, when this is activated, what is going to happen? Bottom line is that the foreign cell, if it's a bacteria cell, whatever, is basically going to be lysed. It will be ruptured. That's the purpose of it. So it is part of this secondary response to a foreign invader trying to help protect you. It's that it will kill whatever that foreign cell is. It has to go through a series of steps. It can be activated in one of 3 different ways. I'm not going to ask that you know the different ways, just that you are aware there are multiple ways it can be activated. Now, one thing to keep in mind when this complement is activated, indirectly what it will do is it will also trigger the inflammatory response, and it can cause fever. See several different things interacting together. This is just a case study. And so there's 3 different ways that complement can be activated. All 3 of them will lead to the same end result, which is where you're going to see all of these different proteins. It's often referred to as a cascade. In other words, you have activation of one protein, that's going to activate something else, in this case something to break apart, and then it's step 3 and then step 4. It has to occur in this specific order. And so this shows a little bit more detail. Once again, do you have to know the names of all these different proteins involved? No. All of this is the complement, sometimes called complement cascade reaction. But I say if you stop right here with the C4, the rest of it will not progress on. So it has to occur in this order. So once complement is activated by any of those 3 methods, like I say, you've got these proteins, you go through all the step, activating each step along the way. It's kind of like, okay, Mary has to break this and tell Joe, and then Joe and Mark have to get together over here, and then they have to tell Sally, and then Sally gets in the group, and it has to occur in this order. What is the bottom line that's going to happen? Is that it's going to form a complex. And that complex right here, this membrane attack complex, this structure right here, it's kind of like it's poking a hole. It's like somebody sticking a straw in a water balloon. If you have a water balloon and you poke a hole in it, the water is going to come out. Well, in this case, if you poke this hole in the surface of any cell, then what's going to happen is it's number one, as you can see, depending on the particular environmental concentrations, things of water, salts, etc., the environmental conditions at that time, water is either going to flow in or it's going to flow out. And also, we'll see later where complement is going to help activate responses in it. This is showing on the slide several of these complexes of this membrane attack complex. Another thing that's considered in the second line defense is the body's inflammatory response. The inflammatory response, this is once again non-specific, so anytime there is type of tissue damage, this will be activated. There are 4 main signs: redness, heat, swelling, and pain. You typically have increased blood flow to the area. That accounts for the redness. You have swelling because the capillaries in that area tend to become more leaky, if you will. So you have increased fluid into the area that leads to the swelling. Blood does have heat to it, so that's why it gets warmer to the touch. And all this extra fluid is pushing against your nerve endings, and so they're pinching. You're going to feel it. Oh, this hurts. Why are you increasing all of this blood flow to the area where you've had tissue damage? Because you want all of those cells that can carry out phagocytosis, you want macrophages to come to this area. You need to have them there to be able to search and find any of these foreign invaders that have been able to penetrate through the damage here. So you can see just on this picture here, you've got your injury site here. These cells here would have released chemicals that attract the macrophages to this area. As I said, your blood vessels become what we call more leaky. It allows for substances to be able to leak out so those macrophages can come and start engulfing all of these foreign invaders. Acute inflammation, this develops very quickly and it's short-lived. Usually this can be very beneficial. Like I said, it's the way of getting the phagocytes there. It's a way of, let's start repairing the damaged tissue. We need to clean up any of the damaged cells, get rid of them, so the healing process can begin. Get rid of any of the foreign invaders. Chronic inflammation is long-lasting, and this can actually cause damage to the tissue that can lead to types of diseases. So this is just showing how during the inflammatory response, like I said, the capillaries here, they do dilate, so you have more blood flow coming through, and then you have more of these mediators like the phagocytes being able to leave the blood supply, go into the tissue, and do their job. And then part of that, like I say, is going to be in the complement activation process, you are releasing certain chemicals that will response as well. And I already mentioned how the blood vessels, we're talking about increased vascular permeability, we're talking about how the blood vessels are going to dilate and allow more release. Here's a monocyte that is leaving the blood vessel, going into the interstitial space, so they carry out the phagocytosis of the foreign substance. So this is just another diagram showing step by step how you've had your injury, you're releasing chemicals, attracting. Now the macrophages are these cells right in here, and you're starting the whole repair process. So some of the chemicals that are involved with inflammation or fever is classified as body temperature over 37 degrees C, which is 98.6 degrees Fahrenheit. There are certain chemicals that are often released when you have an infection that trigger the hypothalamus is in the brain, and that is what is controlling your temperature regulation. It's kind of like the thermostat for the body, and what the pyrogens do is they reset your thermostat. It's kind of like you going over in the wintertime saying, oh, it's a little chilly in here, let me turn up the heat. That's basically what your pyrogens are going to be doing. There's different types of pyrogens. Sometimes you are producing complexes that will trigger this. A slight temperature is actually good for you because in protection or defense mechanism is that you have a wound, those chemicals will, up in the hypothalamus, will reset the thermostat, if you will. And then this will do several different things, ultimately leading to increase in the body temperature. Slight increase in temperature can increase the effectiveness of those interferons, which we see with viral infections. It will actually inhibit the growth of some organisms. It also oftentimes will trigger the liver to tie up some of your iron, which some bacteria need for survival, so that becomes no longer available. They cannot survive without it. It actually increases the effectiveness of phagocytes, and it will speed up the tissue repair. If the fever goes too high, that is not going to be a good thing. So this is a summary showing the first line of defense, those chemical and physical barriers. If the pathogen is able to get past that, then here's what we have for our second line of defense. We have all of these components in our little bag of tricks to try to combat whatever the foreign substance was that was able to penetrate the first line of defense.